This is my third A-level OCR biology video on Unit 2.6, and previous videos we looked at mitosis, meiosis and the cell cycle, and finally we're finishing off this chapter by looking at the more boring bit but the easier bit on cell differentiation. So hopefully I can get through this quickly, hopefully in about 5 or 6 minutes. So let's get started straight away. So we've got animals. A zygote is what we get when we get two haploid cells joined together, so our egg cell and our sperm cell, and this zygote then undergoes mitotic divisions, producing embryotic stem cells which altogether form an embryo. And these embryotic stem cells can then differentiate into different cells. So the way they do this is they can have different proportions of organelles. Certain genes can be switched on and off, the cell shape changes, and the overall contents within the cell changes, such as erythrocytes and neutrophils. So erythrocytes are small, by the way these are red blood cells, they're small, they've got a large surface area to volume ratio, they've got a bioconcave shape, so they can store lots of hemoglobin, therefore oxygen association. They're flexible, so they can move through very small capillaries. They've got no nucleus, mitochondria, endoplasmic reticulum, so they can store even more hemoglobin, as much as possible, and very little cytoplasm. So basically, erythrocytes just want to store as much hemoglobin as possible, so they can carry as much oxygen as possible, and they're adapted to be as small as possible, have that large surface area, so they've got fast rates of diffusion. Neutrophils are also produced by bone marrow cells, which are stem cells. However, they're completely different. They're twice the size. They've got a multi-lobe nucleus. So they undergo chemotaxis, attracting them towards chemicals. They can ingest bacteria and some fungi. So they both come from the bone marrow, but they have completely different properties and properties because they come from because of this differentiation. Another specialised cell is a spermatozoa, so you probably know them as sperm, and so did I until I did A-level biology. They've got many mitochondria to move their undulopodium, which is their tail. They're small, long and thin. They've got enzymes in the head of the sperm, and they release those enzymes to kill off other sperm. They've got spermicide properties, and also to enter the egg cell. And they're found in the acrosome, which is this purple bit here. Then we've got this haploid nucleus, which is half the genetic material. They're made by meiosis, and they've got lots of these mitochondria to move this undulopodium, long tail. Okay, so that's a sperm cell. Another little specialised cell we can look at humans is epithelial cells. So they are flat and squamous epithelial cells. They all tightly packed together. Some can have cilia. Okay, we'll look at a tad more epithelial cells when we come to look at tissues. Now let's move on to plants. So plants have their own specialised cells, just like animals do. We've got this waxy cuticle, we've got upper epidermis, a palisade mesophyll, xylem and phloem in this vascular bundle, which I spoke about in Unit 3.3. They've also got this spongy mesophyll layer that I'm drawing on now, and as well as that, they've got a lower epidermis, guard cells, and stomata, or one stoma is drawn here in between them all. So they're all different cells, they're all differentiated. So let's have a look at the palisade layer. So the palisade layer is tightly packed together. They're long cylindrical cells. They pack together, little space in between them, so water cannot escape through the upper epidermis. they have huge amounts of chloroplasts and a large vacuole which pushes the chloroplast to the cell periphery, the outside of the cell. They've got cytoskeleton threads and motor proteins which can move your chloroplasts to the areas with more light. So they're just trying to make photosynthesis as efficient as possible. Then we've got our guard cells, and they use active transport of potassium ions to lower the water potential within the cell. So water enters the guard cells, makes them turgid, helps them open, then the stoma open. So when the stoma open, or stomata, you get air moving in, so carbon dioxide can get in, water can get out, transpiration streams can occur, and that's covered a lot in Unit 3.3. So is root hair cells, but another adaptation root hair cells have is these hair-like projections, giving them a huge surface area to volume ratio. They actively transport mineral ions, which helps lower the water potential of the cell again, and shift water into the roots. Okay, so this page is a huge number of differentiated cells, and the ones I've spoken to you about are the one the textbook gives me, and I'm guessing the one OCR wants you to learn. So now let's look at some tissues and organ systems, and I really want to whiz through this one just as quickly, if not quicker. So we first got epithelial tissue. So we've spoken about epithelial cells, but epithelial tissue has no blood cells, they're closely packed, they're ciliated, and can be non-ciliated. We've got short cell cycles, so they're always dying. They've got roles to protect, I can't read my own writing, absorb, filtrate, excrete, and secrete. Okay, look into them a bit more if you want. I don't think it's something you're going to get tested in too much depth on. Connective tissue has a extracellular matrix with collagen, elastin, and polysaccharides which trap water. It's made to be flexible to withstand weight, and we see connective tissue in blood, bone, cartilage, tendons, okay, all flexible things which can withstand pressure. Okay, so next we have cartilage, and there's three forms of cartilage. So you can have hyaline cartilage, which forms embryonic skeleton, covering the ends of long bones in adults, joining ribs in the sternum, found in the nose, trachea. It forms the C-shaped rings of cartilage in the trachea. You can have fibrous, oops, I did not mean to do that, fibrous cartilage, which occurs in discs between vertebrae and the backbone and the knee joint, and elastic cartilage, which makes up the outer ear and the epiglottis, which is the flap that covers over the larynx when you swallow. Another specialised tissue is muscle tissue, so there's three forms: skeletal, cardiac, smooth. Not going to go into any more detail than that. Epidermal tissue provides a protective covering, and now we're looking at plants, by the way, of outer leaves, stems and roots, so pretty much protective functions of this tissue. Vascular tissue can be xylem, phloem, cambium layer, all that vascular bundle stuff in Unit 3.3. Meristematic tissue, which is more like stem cells. They've got thin walls, they are able to differentiate into any sort of cell. They've got no chloroplasts, small vacuole, and they divide by mitosis. They're found in root nodules and shoot tips, root tips, shoot tips, and in the cambium of vascular bundles. Okay, that is all of the tissues I'm going to show you here. One thing I've just been lazy and just put in a picture of this textbook, and if you can't read it, we've got plants and their organs and their systems and their roles, and animals with some organ systems. So we've got the leaf, which carries out photosynthesis. We've got the root, which is for anchorage absorption of mineral ions and water, storage. Stem is for support, holding leaves up, transportation of water, transportation of products, storage of products, and the flower, which is for sexual reproduction. In animals, the digestive system, and that's a list of all the little organs contained in it, and that's for nutrition. Circulatory system for transport, respiratory system for breathing, gaseous exchange, urinary for excretion, osmoregulation. I could list all of these off for you. If you want to look at them, please feel free to pause the video and look at them now, but I'm going to skip it right now to the final bit of the chapter, which I can't wait to finish because this is pretty boring to talk about, is stem cells. So stem cells are undifferentiated cells capable of becoming any type of cell, and we've just seen all those types of cells they can become. We've just looked at all the various tissues, all the various cells, and originally they all come from one type of cell, a stem cell, okay? And these are pluripotent cells, so that which literally means able to make many cell types. So they undergo mitosis and can express all of their genes. So they just contain all of the DNA and are able to differentiate into anything. And some example of some stem cells are the embryo, the umbilical cord, the umbilical cord blood. They can be found in adults, within bone marrow, muscles, brain, blood, all sorts of tissues. And they can also be lab developed, which is a huge thing they're trying to increase. And there's so much potential in using this research, and we can use it for bone marrow transplants. It can save people's lives, especially if they've just been suffering with blood cancer, drug research, developmental biology, so studying how cells develop, and trying to find out if they can extend the capacity of embryos have for growth, tissue and repair in later life, used for repair of damaged skins. They can help fix disease. If we can produce stem cells to produce new nerve tissue, it can help treat Alzheimer's, Parkinson's diseases, horrible diseases where people lose their memory because of neuronal issues, the tissue sort of degrade. We can try and repair that. We can also replace tissues, so damaged tissues can be replaced, or like bone marrow which is gone faulty, we can replace that with transplanted bone marrow. And we can also use regenerative medicine, which is when stem cells are used to populate a bioscaffold of an organ and then directly used to develop and grow in specific organs for transplanting. So we obtain the patient's cells, reprogram them to become iPS cells, and then use them to make such an organ. There will be no need for immunosuppressant drugs. And an example this textbook put in, if you don't read the little blue boxes that these textbooks have, just to sort of summarise what stem cells are capable of. In 2013, a 2-year-old girl was born without a trachea. Obviously was not 2 when she was born, but she was born without a trachea, and when she was 2 she received an artificial trachea grown from stem cells from her bone marrow. So they took stem cells out from her bone marrow using a needle, and in a lab seeded them onto a collagen scaffold, allowing them to multiply, create a new windpipe, and when they implanted this trachea, it acted like a normal trachea, and it sent other cells to the trachea which spread out and sent other correct tissues to grow on the inside and outside of the tube, and it just acted like a normal organ. Obviously, as the bone marrow was taken from herself, she did not need to take immunosuppressants as it did not get rejected. But that is all of Unit 2.6. If you've not watched my other videos, I advise you do, just flicking through all what I've done here. It's a hard topic to get your head around, so re-watch videos, watch other people's videos. Best luck in the exam.